Hi, I'm Emily Wu, and today I'm going to talk about the role of glycoproteins in adhesion. So, cell adhesion is important for forming tissues. It's involved in the three different types of junctions: tight junctions, desmosomes, gap junctions, and other junctions used in humans. Some examples are epithelial cells forming the lining of small intestine and hepatocytes forming the liver organ. So cell adhesion is mostly mediated by cellular adhesion molecules, also known as CAMs. They're mostly found on plasma membranes, and they may be connected to the cytoskeleton. There are two types of adhesion: homophilic, where cells of the same type bind together, and heterophilic, where cells of different types bind together. So there are 5 classes of CAMs. The first one is cadherins. They are calcium dependent, and they are mostly involved in cell-to-cell adhesion. And you can see that here with the E-cadherin, or epitheliocadherin. They're involved in tissue differentiation, and they mostly do homophilic adhesion. So you can see 2 cadherin proteins binding together. There's also the immunoglobulin superfamily, which are calcium independent. They involve nerve cell CAMs, and which have long chains of sialic acid, which is a type of sugar that binds together. So you can see that here. They mostly do homophilic adhesion. There are also selectins, you can see an example here, which are calcium dependent. They're involved in leukocyte extravasation, where the leukocytes move through the tissue, and this involves specifically P-selectin. So the leukocytes move through the tissue by binding to other cells and forming and breaking bonds easily. So that's what selectins do. Mucins are calcium independent. You can see that here. They are components of mucus. So mucus protects epithelial cells from infection, dehydration, and injury, and it aids the passage of materials through a tract, so like mucus in your throat. Integrins are the final class, and they mediate cell matrix interactions. So they connect the extracellular matrix to the cytoskeleton of the cell, and they can bind extracellular matrix proteins. So one example of cadherins are that they work in adherens junctions, which is a continuous band of cadherins which connects the lateral membranes of epithelial cells. So you can see in this diagram, the epithelial cells are bound together. They're located near the apical surface, so near the top of the cell, and they contain alpha and beta catenins, which link epithelial cadherin in the plasma membrane to the circumferential belt, which you can see here, which is composed of actin and myosin filaments. This functions as a tension cable to internally brace the cell and control its shape. And you can see in this diagram how the cadherins bind to each other. So one problem that might occur if you have cadherin loss is tumor cell metastasis. So that's when the tumor cells start spreading throughout your body, and it's correlated with a loss of cadherins on the cell surface. So in this case, the tumor cells won't stick together. They'll adhere to each other less avidly than healthy cells. So right now, currently, scientists don't really know why this happens, but there are 3 potential mechanisms. Epithelial cadherin regulates beta-catenin signaling. So if this goes wrong, then it may occur. Then this may occur in tumor cell metastasis. Epithelial cadherin also inhibits signaling during mitosis through growth factor receptors. So you can imagine if it's not working, then tumor cells may multiply easily. The links between cadherins and molecular determinants of epithelial polarity have been found. And epithelial cells have a polarity in that they have an apical area and a basal area. And so without this polarity, they may turn into tumor cells. So one study that's been recently done was how Zika viruses infect Sertoli cells. So Zika viruses are associated with microcephaly in babies or babies with small heads. And they found that this virus can be sexually transmitted even months after clearance of viruses from the semen. So months after there's no viruses found in the semen. And this suggests infection of the seminiferous tubules where sperm is produced. And inside seminiferous tubules are Sertoli cells, which form tight junctions together, and they form the Sertoli cell barrier around the spermatozoa. And they mostly act as a support in the seminiferous tubules. So the scientists wanted to know what is the mechanism of the Zika virus entry into the Sertoli cell barrier. And they found that Sertoli cells were highly susceptible to Zika virus as compared to the closely related dengue virus. So Zika virus definitely had a different mechanism of entering the cells. They found that Zika virus infected macrophages exposed Sertoli cells to inflammatory mediators. And so this resulted in the degradation of the ZO1 protein, which is involved in the tight junctions that form the Sertoli cell barrier, and that increased the Sertoli cell barrier permeability. So the Zika virus was able to enter the seminiferous tubules. So the scientists were able to conclude that Sertoli cells are a great possible therapeutic target that they should do further research into to clear testicular infections. So my question is, what are some other diseases associated with the malfunction of various cans? Thank you.